I have to say I've had several fellows and graduate students from Europe over the years, and many connections in Europe. So for me, Paul, it was an honor to be invited to be an advisor for the concept of the NEUROGUT Training Institute. And now to be invited back at the last phase of it is also an honor. So I thank you for that. Probably the philosophy for fellow and graduate student training originated, or certainly from my concept, originated from something we call the Little Brain, Big Brain group. The Little Brain, Big Brain group met for the first time on September 7th through 9th in 1989, 2 days ahead of the International Gastrointestinal Motility meeting in Gmunden, Austria. I'm not sure the idea originated mainly with Michael Sheeman. I think you can probably pick Michael Sheeman out of this slide. There. What's that? So anyway, Michael's idea was at the Neuromotility, International Motility Society meetings, the big guys dominated. And also Michael had noticed how many of us more senior mentors and advisors developed some animosities in competition with other advisors, and really sometimes that would spill over to trainees. And Michael thought this was not a good idea. And so in 1989, the first meeting of the Little Brain, Big Brain group was held in Munich. And the old idea was for fellows, this group of trainees, to get together, argue as much as you want. We never ended a discussion session during the meetings that they were invited to. But then when you finish, you go out and you party like mad. And all of these younger people now, and there's some here that will attest to that, are now really quite good friends and continue to be good friends and collaborators. The first meeting was held on the military base on the Sanitates Academy in Munich. This is an Army operation that sets up battlefield hospitals. In fact, in disaster zones, they can deliver a functional hospital within 36 hours. From that, in those days, it may be shorter than that. It turned out that that costs a lot of money, so Michael didn't have a lot of money. He mainly did the organization for this. But my first postdoctoral fellow, his name was Klaus Meyer. Klaus that was 1972 that he came to work with me in Kansas City. So Klaus had advanced to the point of the commander of this Sanitates Academy that set up military hospitals. And so it turned out that Michael, and there were some negotiations, Klaus Meyer set up, allowed us to use conference rooms and allowed the attendees to stay in the barracks with the soldiers and also to eat in the cafeteria with the soldiers. American fellows that were at that meeting, they still talk about the impression of being on the military base for this kind of meeting. So the first meeting, Klaus Meyer hosted that. So the concept here, and this carries forward in the philosophy, it seems to me, of the NEUROGUT Training Institute, even to this day. And I hope you continue that sort of a philosophy. This is an opportunity for me to pay my respects to a very important individual in terms of intestinal motility, gastrointestinal motility, and in Europe during the eras that have led up to what we're doing here now at the NEUROGUT training session and the NEUROGUT meeting. And this was Hans-Jörg Erlein, who passed away on June 1st. Hans Erlein, he spent his life looking at motility with contrast material in the lumen on the fluoroscope, and certainly impressed me to a great extent. One of the ways that watching these motility videos, the only way you can understand gastrointestinal motility is to observe what happens in the lumen as material moves. And it's Hans Ehrlein. It's his videos, going through the videos with him several times for some projects that led me to think about the enteric nervous system as holding a library of programs for various behaviors that we observe there. On the left is, for example, I call this postprandial motility program because this is segmentation mixing kind of movements that are there. I have difficulty with clinicians trying to explain there is such a thing as physiological ileus because they always think of pathological ileus, that is, absence of motility. But there is physiological ileus. Many times you can see the gut is so totally silent. You can take a piece of intestine, oftentimes in an organ bath, and when you block all the neurons, it's neurogenic ileus because continuous activity of the inhibitory musculomotor neurons there. Another one is the interdigestive motor program. You may know this as the migrating motor complex. It's been mentioned here before. That is when digestion is complete and the gut is empty. This program then is called up, begins to run, segmentation stops. Hostile programs in the colon is another example. Power propulsion, we've heard this mentioned as giant migrating complexes, but power propulsion, very powerful propulsive motility that empties long segments of intestine in the small intestine, distal small intestine, and colon, in the proximal small intestine, going to in the aboral direction. So I call that the emetic program or the emesis program. Another one stored in the program library of the enteric nervous system. Now, what really triggered me many years ago, I was involved with some development programs with Ehrlein, and he showed me this video where Ehrlein was a veterinarian, and he had this dog had been brought into him, and the dog had just eaten a meal, but in that meal was rat poison. So what Ehrlein was looking at is he also had contrast material in the lumen looking at motility. And so what you could see is the dog had a meal, and he was in the digestive, postprandial mixing digestive program was running. Then he gives the dog apomorphine, and then you immediately see a change from the postprandial program to the emesis program. Very rapid transport of material from mid-small intestine to the stomach. So if everything is right here, I can show you that. Now, you see, this is one of Ehrlein's videos, and this is digestive motility. See, there's segmentation motility. And then we'll see that when we convert to the emesis program is very dramatic. Let me find. Okay, this is your gut when the postprandial enteric motor program is running. Now you'll see this convert in this region right here of the video. We'll see the conversion then to the emesis program. There it's just about to convert, and there it goes. There it goes. Now when I talk about power propulsion, this is also power propulsion. Very rapid, very strong contraction to the circular muscle. Very rapid propulsion of the contents into the large intestine, and now the dog retches and vomits. Anytime you think of cramping abdominal pain, this is power propulsion. Just as you saw here, but power propulsion in the lower GI, distal small intestine and colon. And this is part of a program that I don't have time to talk about, but everyone's experienced it here. When you get a little food allergy, enteritis, whatever, you get the crampy abdominal pain, then you know that you better get to the restroom because you know there's going to be the explosive watery diarrhea. So, but you see, by just knowing Hans Ehrlein, I could appreciate and begin to think about this kind of a concept. So it was only a couple of years ago, an Ohio State medical student held up his hand and said, "Hey, Dr. Wood, you know what you're talking about here?" He pulled out his iPhone. He said, "You're talking about apps. You're talking about apps stored in the library in the enteric nervous system." touch an app, and the program runs. So from now on, I call them apps, postprandial app, medic app, and so forth. But my point here also is I attribute this to Hans Ehrlein and his initiative and his dedication. And it's not just dedication, he enjoyed this very much. Michael Sheeman will tell you this as well, this kind of the kind of research that he did. What I'll do is talk a couple of legends, and then we'll move on to what research opportunities, or at least I feel, are there. This was in 1976. I had an Alexander von Humboldt Fellowship to work in Germany. I was working in Munich. By then, Klaus Meyer had risen to the point of commander of the Sanitäts Academy. So in the laboratory one day, we got the idea, what would happen if you just take out a piece of guinea pig colon, put it in an organ bath under physiological conditions, and take a fecal pellet, put a little KY jelly on it and epoxy, and put it in the lumen of the colon. And in fact, everyone has seen this, and you see video after video of this. But there's the propulsion. Take a picture every 10 seconds. There's the propulsion of the fecal pellet. But you see what this established was an independent nature of the little brain in the gut. That's an independent, integrating nervous system. And of course, it had to be not connected to anything in the body, but it also block that toxin. Required the enteric nervous system, this propulsion of the fecal pellet to occur. I move on to what I consider to be, for you all are going to be looking for projects. You're going to look for something that's going to so you can make a living, but that you can also advance science and select projects that are going to pay off that way. So I'll just put in 2 or 3 that I think have quite a bit of potential for what you all might want to consider. One of those is stress. What happens if you take a guinea pig like this and you wrap him in tape and put him in a cold room for a couple of hours, and then a control, of course. What you'll find is that cold stress, causes that guinea pig to poop like mad. Here's the control, and here's the fecal output of one guinea pig in response to cold restraint stress. So, and here's the data, the number of fecal pellets here, and the water content of the fecal pellets. And then over here in cold restraint stress, see the water content. Since we're talking about guinea pig diarrhea, there increased water output, and also numbers of fecal pellets. It turns out in this model, if you treat it with selective CRF, corticotropin releasing factor, CRF1 receptor antagonist, you block that. So it's becoming a big deal, the involvement of corticotropin releasing factor, and especially in the little brain in the gut, the way it acts in the gut. Now everybody here, irritable bowel syndrome, every lecture you hear related to functional GI disorders, stress comes in as a factor. And stress comes in a lot of other places. And I'll tell you, there are opportunities there for the various kind of study, mechanisms of stress, or whatever in that relationship. Here's what we know about stress so far, that it impacts GI function in most every animal species. The stress is mediated by the enteric nervous system, by the little brain in the gut. Neurons in the enteric nervous system express CRF and its receptors. Stress stimulates CRF gene expression in the gut. We already know that. And then also in the neurons. Stress elevates excitability of CRF neurons. They fire much more. And stress-induced activation of CRF neurons in the enteric nervous system underlies what I just showed you, the fecal output, stimulation of mucosal secretion, and opening of the mucosal epithelial barrier. The pathways here, the stress points, and this is still wide open to be studied. The hypothesis these days is stress somehow involves enteric nervous system. We've also heard in this meeting, maybe also immune inflammatory cells. CRF, mast cells express CRF receptors. And now the release of mast cell mediators are responsible for calling up a program that includes power propulsion, massive secretion, and abdominal pain. We've heard quite a bit. Well, we heard from the guru of intestinal afferents, spinal afferent from the intestine a moment ago, the person. This is going to continue to be a big area, but I think the big area that's looking very important is not only do spinal afferents take sensory information to the central nervous system and underlies all the sensations we associate with discomfort, intestinal pain, but those spinal afferents also innervate enteric mast cells. And here's an example. Spinal afferents. David mentioned these. I'm going to use the pointer. David mentioned how some spinal afferents innervate neurons in the enteric nervous system, and so we realized this. And so what we did is we have a preparation such as this in the organ bath, and then we stimulate the spinal afferents, but backfire them. Fire them, stimulate electrically here, and so now action potentials propagate back. And what we can show is this also releases mediators from enteric mast cells. So we just use ELISA assay, what's released from a segment of gut like this, and we begin to pick up histamine, mast cell proteases, substance P, calcitonin, gene-related peptide. Of course, the substance P and the calcitonin gene-related peptide, these are neurotransmitters. They're sensory neurotransmitter. Well, one of our chairs here, Dr. Giovanni, has shown already about mast cell mastocytosis, for example, in irritable bowel syndrome. Oh, I need to go back. So what you see, if you stimulate those spinal afferents and you measure stuff that comes out in the bathing media, here's an example. Over here, we get mast cell protease 2. That's a mast cell degranulation product, and you can evoke that with a sensory neuron with an afferent neurotransmitter. CGRP, also sensory neurotransmitter. Capsaicin, activate the afferents with capsaicin, and there's the increase of mast cell protease 2. And likewise, stimulate the afferents, backfire the afferents, and then you block this with the tetrodotoxin. So this is evidence that spinal afferents also innervate mast cells. Same thing in human intestine. Get the human intestinal segment, small and bowel, from the gastric bypass patients during Roux-en-Y gastric bypass setup. Same story, and in small and large intestine. Think a lot about histamine also as one of these mediators. So what it looks like, and the reason I think this is a good area to think about in terms of future research, certainly kind of research that David Grundy told us about as well. It's what it looks like. What it looks like is, I want to go backward. The way it looks is if spinal afferents. Here's the spinal afferent. Obviously takes information to the spinal cord and on to the central nervous system. But these afferents also seem to give off collaterals that innervate enteric mast cells. Now the significance of this was evident in Dr. Grundy's lecture, in that these spinal afferent terminals express receptors for mast cell mediators. Receptors for such as adenosine, histamine, mast cell proteases, all act. And I think David has shown most of his students that they also fire these afferents. But you see the ramifications of this is with these collaterals, anything that fires these receptors is from coming from mast cells. Now, if a collateral activates mast cell, releases these mediators, that sensitizes this terminal. So a receptor, whatever its modality, mechano, chemo, thermal, whatever, is going to be sensitized. The situation here then makes this look as if there's some kind of a negative feedback or a positive feedback that amplifies sensitization of the afferent. You see, there's the receptor, there's the afferent fiber. These afferent fibers, we know they innervate enteric neurons. We fire enteric neurons when we stimulate them. It also releases mast cell mediators. This is a positive feed-forward loop. It's going to sensitize the afferent. Afferent becomes more sensitized, fires higher frequency back to the mast cell. And what we don't understand yet is why we can see how this can lead to hypersensitivity, but what terminates it? How long will this sort of thing run? And that's where we are. We don't understand it. We've got this model, but there's much work that could be done with that. I think one of the very, very exciting areas these days is transgenic animals and gene insertions and mechanisms for studying the enteric nervous system with so-called optogenetics. This is going to be hot in the next few years. Here's an example from Nick Spencer and Hong-Chin Liu. Both these guys were little brain, big brain fellows in their day. But what can now be done, and what they've done there with Cree-Lox technology, is label neurons. For example, in this case, this is a neuron, a so-called calretinin neuron, which we know are cholinergic neurons in the little brain in the gut, and also then with Cree technology and with gene insertions for markers such as fluorescent yellow protein, they can now mark what we call an interneuron that uses retinol, that expresses retinol. That's the neurohistochemical identification of a retinol neuron. We know those are excitatory neurons. So now what they can do is they can put rhodopsin channels into specifically into that kind of neuron, and then activate those neurons again with blue light. And now then they also have techniques now for inserting genes for toxins, neuronal toxins like diphtheria toxin. So the ability to take out a particular specifically identified neuron in the enteric nervous system and then ask, what does this do to motility? This is an example. From Nick and Hong-Chin Liu. It's a chapter, the 6th edition of the Physiology of the Gastrointestinal Tract will be coming out on January the 2nd. And so Hong-Chin and Nick, I asked them to put this kind of stuff in the chapter in the Physiology of the GI Tract. So that's where I got these illustrations. You see, here's a neuron with expressing green fluorescent protein. Here's the neuron in this preparation. It's expressing calretinin, just calretinin. We know calretinin are cholinergic neurons, so if we do immunohistochemistry with green-yellow protein marking the neuron, see a lot of choline acetyltransferase expression. This is a marker for a cholinergic neuron, and that tells you that we've hit the calretinin neuron. For NOS neurons, which are the inhibitory musculomotor neuron down below, they're labeled with green fluorescent protein, but they don't express CAT because they're non-cholinergic. And what is I've never believed this, but now then with channel rhodopsin inserted in a retinal neuron, now you do the same study Klaus Meyer and I did in Munich in 1976. Do that same study now. Here's the mouse preparation rather. Expose it to blue light, shine the blue light, and you evoke propulsive motility in this preparation. This is the video, Everybody has a video of the fecal pellet in the colon, in the guinea pig colon. This is a video of the same thing, except with activated blue light and channel rhodopsin. Whoop. Triggers the propulsive motility. Now the next slide, the next images are with tetrodotoxin, which blocks that. But the other thing I want to watch for is with the tetrodotoxin, also with the fecal pellet experiment. With tetrodotoxin, you see hyperactive motility all of the time. That's because the inhibitory muscular motor neurons are continuously active, and that's also what accounts for what I call physiological Ilias act. I suppose we want to leave this run, she said, because there can be some kind of problem if I terminate the video too early. This is with tetrodotoxin. Okay, I think finally, let me tell you, in terms of what time were we supposed to stop?
We should be
stopping. What I wanted to finish with is the opportunities for you all also for translational motility that's also neurogut related. One of those is putting genes in diseased individuals to change a mutated gene. And what I've just shown is here is the advantages of using adenoviruses to put those genes in there, and for several neurodisorders, this is already being done. Another is this idea in irritable bowel syndrome that the explanation for many of the symptoms is that these individuals, for some reason or the other, are developing anti-enteric neuronal antibodies that are destroying the enteric neurons. This happens to be from a chief of neurology at Georgetown University. He called me up and said, "Jack, I'm getting IBS symptoms, postprandial diarrhea, postprandial pain. What do you think?" And so I said, "Well, send us a blood sample." And we incubate that blood sample with enteric neuron. And sure enough, he has big reactions to his enteric neurons. Those antibodies are binding to his enteric neurons and apparently lysing the enteric neuron. So that's another big area. It turns out that those antibodies in our studies, those antibodies only identify 3 antigens in the enteric neurons. The one that's interesting there is the RO5200 antigen, because those are all nuclear antigens. This is one that's associated with dry mouth and Sjogren syndrome. So it makes us wonder, and this is the reason I say for research, consider this, because these are also the kind of parasympathetic neurons that control the salivary gland and so the periphery. So that's what I wanted to tell you. Big brain, little brain offers many opportunities, and I hope you all pursue those and